Good afternoon, and thank you for having us here speaking. I'm Olga, and Cecilia is going to take over afterwards. So we are focusing on the utility of long-read sequencing in myeloid neoplasms. And I'm going to skip over the overview. So we saw the potential of using this long-read technology as it may overcome some of the limitations of traditional next-generation sequencing, and also in order to sequence some hard-to-sequence like regions. Also with fusions. Fusions are in some situations, a rapid detection of fusions in AML is critical both for the election of treatment, or in some cases because patients may be quite sick, and it's important to know what is exactly the disease that they are having. And also, this detection of specific breakpoints may assist on personalized molecular residual disease or detection, and on the study of the clonal heterogeneity of the disease. For that, we decided to go with the enriched protocol using CRISPR-Cas9, and it doesn't require amplification, so that was really attractive. And also, it would give us enough. We thought it would give enough coverage, and with a decent amount of or achievable amount of starting specimen. So we designed guides that would target all the genes that are involved in recurrent translocations in acute myeloid leukemias. For the analysis, we partnered with our collaborators at the University of Washington that had developed the Biodepot Workflow Builder, which is a graphical tool to create and reproducibly execute bioinformatic workflows. So we adapted this workflow for processing Nanopore data. In this platform, each computational task is encapsulated in a Docker container, so that helps enhance reproducibility, and the input parameters are just chosen with a drop-down menu. It supports graphical outputs, including IGV, and it's a cloud portal that users can launch with pre-configured GPU-enabled virtual machine instances. So in the example before, which was published in 2021, we see that with the use of this platform with GPUs on the cloud, we achieve a 29-fold speed up in analysis time with a 93-fold reduction in cost. This is a graphic example of how the platform works. On the left-hand side, well, this is two different workflows. On the left-hand side, you have different packages that are represented by the vignette, so you can drag and drop them on the panel, and you just create your workflow, and just hitting the play button you can see how it goes through all the different packages and get the results at the end. The GitHub address or the website is at the bottom. So with this platform or with this analysis and assay design, we achieve these are some of the timelines for analysis of the data. We decided on 3 fusions was enough for diagnostic, and that's based on other platforms that we routinely use in the laboratory for patient diagnostics. And so the dark blue block is the time we spend from the receiving of the blood specimen to the DNA extraction. And the light blue would show the library prep time. The orange bars in these 3 examples shows the time that we needed to have the sequencing going in order to achieve the 3 reads, or the 3 reads containing the fusion that we were expecting. This is a retrospective, or we archive samples. And the green block just shows the time spent in base calling, alignment, and the Fusion Finder workflow. This is just an example of 6 cell lines, and the average time we needed to sequence and analyze the data in order to get the 3 fusions, which was 42.75 minutes on average. And on the bottom is 11 primary specimens that we achieved the 3 fusions with an average time of 188 minutes of sequencing and analysis. The bottom, like the bottom writing, just shows the average number of reads needed for in each one of the cases, 11,000 for cell lines and 10,000 for primary specimens. And now Cecilia's going to show you the clinical cases.
All right, thank you. Okay, so for my section, it's going to be more clinical and kind of what we were trying to achieve with this, and really from a global oncology perspective, what is the crux of the problem from a low and middle income country perspective? So the Fred Hutch, we have a connection with Uganda. We have a Fred Hutch Cancer Center in Uganda. So this is a very front and center, and I am a faculty in our global oncology program. While 20% of cancer deaths are worldwide, why is it that 70% of cancer deaths are occurring in low middle income countries? And that's because in the red and orange regions, there is a severe lack of resources. That's where the deaths happen, but there's also a severe lack of pathology and oncology resources in these regions. And key factors that have been identified include earlier diagnosis and also up to 25% of low-income countries actually even have access to pathology diagnosis. So this is where we thought that Nanopore could really make a difference. And specifically here, I show you two diagnostic entities which has a critical need for rapid diagnosis. One is acute promyelocytic leukemia. This is characterized by 1517 PML RARA. And for these patients, they present in DIC, disseminated intravascular coagulopathy. These patients come in critically sick. However, if they are diagnosed, you can give them Atra, which is essentially a vitamin A derivative. It's a pretty benign drug, and that can really save their lives. Second is chronic myeloid leukemia. And this is actually a very easily treatable disease where these patients, if they are given a tyrosine kinase inhibitor, they can actually have a normal lifespan. So really important to be able to identify and be able to provide these patients with treatment. And so we identified these two diseases as kind of the first things that we wanted to find patients that have these diseases and confirm and diagnose them. So in the newest edition of the WHO, the 5th edition, we've kind of gone through an iteration where we now try to have a cytogenetic and molecular classification system. And there is another classification, there's a big hoopla in the heme path world. The ICC also went through the same strategy. And so here is defined a number of molecular and cytogenetically defined entities. And so if we can actually have an assay that targets these and we can identify the patients, then perhaps we can actually guide these patients into a targeted therapy strategy much faster. And so I'm going to go through 3 cases in the next few minutes here. So I'm going to go kind of quick. Our first case is a chronic myeloid leukemia patient. This is a 62-year-old man. He has a long-standing history of CML. However, he was treated with imatinib. That's a first-generation TKI. He stopped for a year, and he came back with a very high white count, not surprising. He actually progressed into acute phase and was switched to a second-generation tyrosine kinase inhibitor, dasatinib. And he actually progressed into cytogenetics as well. And so at that point, we were able to perform long-read sequencing on him and actually showed what the specific translocation was. And also, by Sanger sequencing, we were able to identify what the actual breakpoint. One of the problems with BCR-ABL is the breakpoint spans over an 11,000 base pair region, so it's really hard to actually create a patient-specific MRD assay. And so we were able to do that with long-read sequencing by Nanopore. And so the follow-up here is that you are able to identify the precise breakpoint in this patient. Precision diagnostic is possible. And this patient ended up developing cardiovascular disease over a 10-year follow-up period, and he was eventually lost to follow-up. Second patient is acute myeloid leukemia patient. This is a 39-year-old man. He actually presented the ED with sore throat, hematuria, and was discovered to have an extremely high white count, 156,000. And showed 90% blasts. And so cytogenetics did show translocation involving the KMT2A gene. However, when we did long-read sequencing, it was really interesting. We actually developed the assay not against the 911 translocation. We actually developed it against a different translocation. But it's really interesting. It's a good thing that our design actually doesn't need both sides. We just need one side to be able to capture. And so KMT2A translocated AML is actually very interesting because this is one of those genes where it's very promiscuous and it can have a number of different partners. And so we were able to pick up this translocation. This patient follow-up was that patient did receive chemo, was followed up with an allogeneic transplant, went into complete remission, and is alive and well. Our third case, now this is a fun one. And some of you might have read the paper on this one. It's a 39-year-old man who presented with thrombocytopenia, easy bruising in DIC. So if you remember the slide on APL, we thought, hey, this could be an APL. It was really interesting. By morphology, this patient looked like he had an APL. They looked like promyelocytes. The flow cytometry also matched, was HLA-DR and 34 negative. However, karyotype showed an isochromosome 17 and not the classic 15-17 translocation. StatFish also did not show the classic PMO-RARA fusion. Long-read sequencing showed a genomic insertion between exon RARA, exon 2, and exon 3 in the intronic region. That's a long intronic region. So normally, by any other method, you would never have been able to detect this. Only by nanopore sequencing could you have detected this intronic insertion. When we sequenced this out, it turned out, and kind of blasted it, turned out to be a viral genome, the TTMV virus. And we sat on this for several years because we were waiting for the second case, right? Because you can't ever publish on one case. And in the meantime, an Italian group published 2 cases. And so we also looked at, and I wish there was a little laser thing here, but when we actually sequenced out the cDNA, it was TTMV-RF2 promoter that was connected to the intronic region and connected to RARA. So it did create an RNA product. And just like what the Italian group also produced, it actually showed the same thing. So it was an extremely fascinating case. This is the first time, well, we're the second group, but this is the first time that we've ever made a connection between this virus actually producing a product and instilling leukemia. So a little bit more about this virus. It is a well-described virus, infects people chronically, and has been described in a number of diseases, but has not actually been described in inducing acute leukemia. So pretty interesting. So the follow-up here is that patient did fail initial 7+3, that's standard chemotherapy. So induction failure, but he was followed up and had to be escalated, and then he actually had to finally also be added with decitabine and venetoclax. So now he's alive and well, 6 months, as of 6 months ago, his last follow-up. And so some conclusions here. You know, what Olga had described earlier with that 3 fusion experiment, we were trying to really push ourselves. And we went back and looked at the data, because as you know, with Nanopore, you can package out the timing. So we were trying to push ourselves. What is the fastest that we can actually get an answer? And that's really what we're thinking about in the clinic. It's like, what is the fastest that we can confirm a leukemia and actually help patients out? And so that was where, you know, we're going through the pipeline. And if we were doing this in real time, what can we really do to help patients out there? And the BWB platform is really nice because it is a cloud-based platform that we can actually get things in real time. And fusion reporting, as you can see, is extremely important now that we do have like more than half of our entities are defined by that. Okay, that's the disclaimer that we have to put in. And thank you.